Okay, so welcome to the exciting session on histone variants and nucleosome dynamics. And the first talk, the keynote speak from Steve Henikoff. So let me spend just a minute on introducing him. So Steve is at Fred Hutchinson Cancer Research Center, and he received his BS in chemistry degree from University of Chicago, working on biopolymers. Afterwards, then he moved to Harvard University, worked with doctor students, worked with Matheson on RNA biology, and then afterwards he went to University of Washington as a postdoc fellow, working on now epigenetics, position effect variation in Drosophila, and he then has been at University of Washington, Seattle since, and now he's full professor and investigator at HHMI since 1990 at University of Washington and Fred Hutchinson. And he has made numerous contributions in the research field of epigenetics and also in biotechniques. So he discovered, one of the major contributions, discovered nested genes, and he, together with his wife, invented Nucleosome algorithm, and also invented tiling and SIFT methods. And he has published more than 250 papers, and many of which have important impact papers in histone variants and alternative chromatin structure. And he has been recognized by many honors. Some are listed here. He, as I mentioned earlier, he was selected to the Howard Hughes Medical Institute since 1990, and he was elected to the National Academy in 2005, and he sits on the advisory board of many academic and industrial institutions, and he is also on the editorial board of many journals, and he is the co-editor-in-chief of Epigenetics and Chromatin, a new journal. And so without further ado, we're going to hear from Steve his exciting latest results. Let's welcome.
Thank you, Ramin, for the introduction, and I'd also like to thank Yi, Ramin, and Rob for the invitation to give this talk. The session is entitled Histone Variants and, is this on? Oh. It's up there. Okay. So the session is entitled Histone Variants and Nucleosome Dynamics. So let me just start out by explaining what the relationship between the two might be. So if you look at this view of the nucleosome, which shows just the histone fold domains, you can see that it forms a tight left-handed spiral that the DNA simply follows around. And there's a central disk of histones 3 and 4, and it's flanked above and below by dimers of H2A and H2B. Now, the bulk of the nucleosomes are deposited behind the replication fork, so they go into newly synthesized DNA. However, there's a set of histone variants that are replication independent in their assembly, and therefore must go into an already chromatinized template. And if we consider those that are universal, there are 4. 2 of them are histone 3 variants, and 2 are H2A variants. Now, to replace H2A with, say, H2AZ that Craig Peterson is going to be talking about in the next talk, you have to partially unravel the nucleosome to make the exchange. Now, to replace H3, which is right in the middle, with H3.3 that we heard a little bit about last night, you have to unravel the nucleosome at least halfway, and we'll be hearing more from Bing Zhu about that in this session. In other words, if we follow H3.3 patterns, we can get a glimpse into nucleosome dynamics as defined by the unwrapping and the rewrapping of the nucleosome core, because the only way you can really get H3.3 in there is to do the unwrapping. So that's the connection. And we've done this for H3.3, particularly in Drosophila, this just shows an example of a gene-rich region. And we see that H3.3 has regions where it goes quite high, and that turns out to correlate to active transcription, as if when polymerase goes through a nucleosome, it disrupts it in such a way that sometimes you get a replacement occurring. But this is steady state, and what we really wanted to do was look at the kinetics. And for that, we came up with a metabolic labeling approach that goes like this. We use an analog for methionine that'll go into all proteins with methionine, and it has an azide group on it, such that it'll go into, when it goes into proteins, we can derivatize it with an alkyne, so-called click chemistry, and that allows us to add a biotin. Now, since we're interested in the DNA that's wrapped around the nucleosome core, we extract chromatin the usual way, and then we wash stringently with urea to remove the H2A, H2B dimers, and that'll also remove essentially all the bound protein. So then we can do streptavidin pull down of the newly synthesized H3H4 core particles. We call this method catch-it. It's the work of Roger Deal when he was a postdoc in the lab. And this shows the catch-it profile for the same region. And I think you can see that it corresponds pretty nicely to H3.3 patterns, although with better resolution because we're looking at kinetics rather than steady state. And what this implies is that our assertion that we're measuring turnover indirectly when we look at H3.3 incorporation is supported by the fact that when we actually look at turnover directly, we see similar kinds of patterns. Now, what about epigenetic regulatory elements? Well, we're working in Drosophila, and these elements are of two types. They're trithorax group proteins that will bind to elements to maintain a gene in the active state, and then there are polycomb group proteins that maintain a gene in the silent state. And if we use chip-chip data from previous work, that for zest, which is a trithorax group protein, and we line up all the zest sites, and then average our catch-it profiles over them, we see that there's a peak of turnover that corresponds to the peak of zest relative to the regions on either side. And when we look at polycomb group proteins, in this case EZ and PSC, components of the PRC2 and PRC1 complexes, we also see a peak relative to the regions on either side. But you'll notice that the overall profiles differ. And what this suggests is there's more turnover at sites where trithorax group proteins bind in general than polycomb group proteins bind. That actually fits pretty nicely with work from Bob Kingston in vitro and Wendy Bickmore in vivo, who showed that the PRC1 complex acts by compacting DNA. That compaction would reduce the turnover. So this turnover might be a marker for the different turnover might be a marker for whether a gene is in the active or silent state. Now, we can also estimate the kinetics of turnover at these peaks. And it's about an hour, which is a short time, but I mean a long time, but it's actually a short time relative to the 20-hour cell cycle. And the implication of that is that at these epigenetic regulatory elements where epigenetic memory has mapped, the turnover is too fast for histone modifications or variants themselves to be inherited. In other words, what I often hear referred to as epigenetic modifications aren't epigenetic at all, really, because they're turning over. There must be multiple erasure events every cell cycle. In other words, it's not like DNA methylation that gets put on at replication, persists stably throughout the cell cycle, and then gets distributed to daughter strands. But rather, there must be some kind of positive feedback loop that is maintaining histone modifications in the same state. Well, this is one way of measuring turnover quite directly, but actually we've been doing quite a bit with another method, and I should point out that it's a method that actually dates back to the 1970s, and we use this enzyme micrococcal nuclease that many of you are familiar with. But micrococcal nuclease is a single-strand specific secreted glycoprotein. So how does it cleave double strands? And what we think happens is DNA will breathe a little bit, and that will allow eminase to cleave one strand, and then it might make it easier to cleave the other strand, and then nibble on the ends until it reaches an impediment such as a nucleosome. And what's often done is to get these nucleosome ladders. This is oligonucleosomes. This is mostly mononucleosomes. This is from yeast chromatin over an eminase series. And what would be done would be to cut out this band here and just to map it to see what the nucleosome landscape looks like. But we realized that eminase will stop at any impediment, such as a DNA binding protein. So we modified the Illumina library protocol in such a way that we could recover fragments as small as about 25 base pairs. And then we did paired end sequencing on them, so we get the size, and then we map and stack the reads. This is an example for this series for yeast. And so we're looking at a particular region, intergenic region of the yeast genome, in which we're looking at the subnucleosomal particles, and we see that there's some substructure there, and it gets whittled down such that we see 3 squarish peaks, each with different occupancy, and each one about protecting maybe 20 or 30 base pairs. And it's stuck in between 2 nicely phased nucleosomes, also showing different occupancy. Now, if we look at a region that we understand much better, this is a regulatory region for the GAL1 and GAL10 genes, and UASG is the site, is the regulatory element that's bound by the GAL4 transcription factor, although these cells are grown in glucose, so there's no GAL4. And as Mark Potashny had previously shown, what we see here is a partially unwrapped nucleosome over this regulatory region. But interestingly, during our M&A series, we're losing that nucleosome, and other nucleosomes remain behind. They're stable, and this one just goes away. In the meantime, we see that there's a subnucleosomal particle that increases its occupancy whenever this gets lost. Notice that these decrease in occupancy, but this one increases. And we know from Potashny's work that this is the risk ATP-dependent chromatin nucleosome remodeler. And therefore, we might interpret this as risk is sitting right next to the nucleosome, causing it to be partially unwrapped. And when you cleave with eminase, the nucleosome will fly off and risk will take its place. So we can observe nucleosome dynamics actually ex vivo using this simple assay. Well, yeast has a small genome, so let's go to a larger genome, and we go to Drosophila. So this shows a histogram of length for total nuclei as well as a salt fraction. So back in the 1970s, again, Marilyn Sanders had come up with a salt fractionation method. And if you use just a low salt, what you pull out is what's been classically called the active chromatin region. You can see that you get a different distribution of sizes here. So for total nuclei, we see a peak at 147. That's the well-wrapped nucleosomes. But we also see larger sizes. That's because of histone H1, which protects more of the nucleosome from DNA, from eminase digestion, and a so-called chromatosome. Well, there's no H1 in the active fraction, so we see a nice peak here. We also see some smaller peaks, and we realized that among those smaller peaks, what we're seeing is RNA Pol2. And so we did CHIP, pulling down RNA Pol2, and we lined up our Pol2 data with data for pause polymerases from Karen Edelman. And what we see is that there's a preponderance of Pol2 from this active fraction that's paused because it's just downstream of the transcriptional start site. 30 base pairs downstream. The footprint is about 45 base pairs. That's exactly where pause Pol2 is. So we're mapping pause Pol2 directly here. So if we look at a particular region of the Drosophila genome, this is a region with 3 tandemly repeated heat shock protein 70 genes. And what we see is there's pause Pol2, as John Lish showed a long time ago. We see some subnucleosomal particles, both upstream and downstream of the genes, and we see a complex nucleosomal landscape. Now we give a 15-minute heat shock, and we see dramatic changes. So we see that Pol2 fills up in the gene bodies. We see the footprint of Pol2 in the subnucleosomal fraction, and we see a reduction in nucleosome occupancy corresponding to that. If we take a close look at one of these clusters, what we see is that these small particles correspond to squarish peaks, just as we saw in yeast, although they're separated from one another. And what we're looking at are DNA binding proteins, which go down with heat shock quantitatively. And this one we know from work of other people, that this is Tata binding protein and this is GAGA factor. So we can do this kind of mapping in Drosophila by just looking at the active chromatin fraction. We're currently doing it with larger genomes as well. So as the depth gets further, we can discern more at single base pair resolution. We also did catch it in order to see what kind of turnover changes we get with heat shock, and we see a very striking increase in nucleosome turnover in gene bodies, which confirms that when the polymerase goes through a nucleosome, when it's actively transcribed, it actually is disrupting nucleosomes, and sometimes you lose it, and that's why you would get H3.3 going in there. Now, there's a limitation to this kind of mapping and stacking, and that is we're not taking advantage of all the length information that we have. And so we realized that we could see a lot more if we could display the length information in a dot plot. And let me just describe the dot plotting approach, which we call Vplot. Let me just describe it for an example of a yeast transcription factor called ABF1. Now, it was known from low-resolution chip data and motif finding that there are about 270 well-determined ABF1 sites throughout the yeast genome. So we lined them all up exactly to the base pair at the site, at the ABF1 motif. And then we're going to draw a dot plot as follows. If we have a fragment that spans the ABF1 site, we take the midpoint of the fragment and we map it on the x-axis as to its position, and on the y-axis as to its length. So that puts a dot within this sector here. If we have a fragment that's precisely cleaved on one side but randomly on the other, that will create this diagonal. So you realize that single cleavages can be detected as diagonals. Likewise, cleavage on the other side will give you this diagonal, such that the minimal protected region will correspond to the vertex of the V, which for ABF1 is something like about 20 base pairs protection, and that's the width of the shadow that you see here. Now, we also have a lot of DNA binding proteins because they're small out here somewhere, and that's what we're looking at there. And nucleosomes are big, and so they map up here, and you can see these nucleosomes up there. Now, we did this for all 118 DNA binding proteins in yeast for which such data are available, and here's some examples. ABF1 and REB1 show these very sharp patterns. CBF1 shows a weaker pattern, but nevertheless a pretty good one, whereas SUT1 shows nice binding because you see the V, but there's no phasing of nucleosomes. Now, we were intrigued by these patterns just by looking at them, because what we realized was there's a couple of odd things. So ABF1 and REB1 show these phased nucleosomes same distance away from the DNA binding protein. So what is it that's causing that action at a distance? And the other thing was, from the footprint, we would basically say, oh, it's 20 base pairs protection or so, but these fragments are really much bigger that we're seeing there. And what we realize that's telling us is that ABF1 is sitting down at a particular site, and there's something next to it that's associated with and blocking from eminase digestion, and it's happening independently on either side, and must be dynamic because sometimes it's not binding and you get eminase cleavage. What would that be? Well, work from Blaine Bartholomew in vitro had shown that the I-switch class remodelers, ATP-dependent remodelers, different from the RISC that I mentioned. The I-switch class remodelers actually photocross-link to linker regions. So we focused our attention on I-switch class remodelers, and there are 3 of them. And when we did CHIP for I-switch class remodelers, we got the same thing for all 3, I-switch 1, I-switch 2, and CHD1. And what we see is consistent with them being associated with ABF1. I'm just showing ABF1 in this case, REB1 similar. And we don't see any of the particles on either side. So that must mean that the I-switch class remodelers are redundantly binding to either side as follows. So to create this kind of pattern, there must be fragments that are protected on either side, but it's probably transient because we're seeing fragments of all different sizes. Now, to get the diagonal, what must happen is that sometimes it comes off, and now the eminase can cleave in there and give you this side, and likewise on the other side. And then the DNA is available now for binding by the DNA binding proteins, and this we see as a dynamic cycle. At the same time it's doing this, it'll keep the nucleosomes from encroaching because they're turning over much more slowly. In other words, the high dynamics of the I-switch remodelers would basically cause a region to be depleted or free of nucleosomes in a dynamic way. Now, if we add up how much I-switch 1, I-switch 2, and CHD1 there is in the yeast cell, there's enough to occupy 10% or so of the yeast genome. So we think that these three proteins are almost entirely responsible for making the nucleosome depleted regions. We know that if you delete them all, then you lose a lot of the phasing. So they do seem to be acting redundantly in that way. I think that the same kind of model will hold very nicely for higher eukaryotes. For example, we've heard about CTCF, and it phases nucleosomes pretty well. If it's bringing in I-switch class remodelers, I think you would maintain nucleosome depleted regions around it. And so it would be acting similar to Rev1 and ABF1, and many other DNA binding proteins would do that. And so these are abundant. The I-switch and the CHD class of remodelers are hugely abundant in mammalian genomes, for example, and I think there could be enough of it to account for maintaining regions free of nucleosomes in a dynamic sense. So we've used this kind of methodology to tell us about what's going on in the nucleosomal landscape, and what I'd like to turn to for the second half of the talk is to focus on our favorite subject, and that is centromeres. So what we're going to do is focus on what's going on at centromeres using these same kinds of methodologies. And I'm showing here a nice figure from a review from Ben Black and Don Cleveland, who point out very nicely that there's been this debate, this intense debate about what is the structure of the centromeric nucleosome. We hear a little bit by way of introduction from Ruming, and I'm sure we'll hear a lot more from Don. But let me just point out that Don is a proponent of this model here, and we're proponents of this model here, which is right-handed hemisomes. And it's based on a few studies, one from Yamini Dalal when she was a postdoc in the lab, and also in her own lab in fly and in human, where we saw arrays of half-sized particles that had all four histones, and also from Taki Furuyama, a postdoc in the lab, who had shown that the particle in yeast in vivo actually wraps in a right-handed manner. So what I'd like to do is go on and show you some of our more recent work that I think can resolve these issues. And we're going to focus entirely on budding yeast, mostly because in budding yeast we know where the centromeric nucleosome is to the base pair. And that is where we know the centromere to the base pair, and that's this 120 base pair region that's necessary and sufficient for centromere formation and function. And it has a tripartite organization. It has a binding site for the CBF1 protein that binds to an 8 base pair consensus, CDE1. CBF1 was actually one of those I already showed a Vplot for, so it's a conventional DNA binding protein. The other is a kinetochore specific complex called CBF3 that binds to the CDE3 element, and this recruits the centromeric nucleosome. And my colleague at the Hutchinson Biggins had previously shown that this is associated with a single centromeric nucleosome in yeast. And so we just apply the method that I showed you to pull down for CSE4, and then we're going to map the occupancy of exactly where is the centromeric nucleosome. And what we see here when we go through the M&A series is a clear pattern. So for centromere 4, we see a shelf over CD1, a shelf over CD3, and most of it is right in the middle of CD2, and there's nothing outside of it. So we see the tripartite structure of the DNA reflected in the tripartite organization of centromeric chromatin. And then if we do V-plotting on the same data, that was centromere 4. This is all 16 yeast centromeres, and they show exactly the same thing. So this is very clean. And here's what the V-plot looks like for all 16 yeast centromeres. So we have a vertex here that corresponds to 120 base pairs of protection. That's protection of the whole region. But we also see these satellite Vs, one directly over CD1, the other directly over CD3. And we interpret that as follows. Because we have three particles here that are very close to one another, there's very little eminase cleavage. So there's occasional single cleavages that we'll see precisely in here and then random on the other. And that's how we can pick it up using a V-plot, which otherwise we wouldn't be able to see. And we see it both for CD1 and CD3. But you'll also notice that there's fading of the V as we go down. And what that implies is that the cleavage causes loss of the particle. So, and we can find those particles actually in the total chromatin. This is the input. We can see the particles that got released. So that shows very clearly that there's 3 independent particles over this site. And that leaves only 80 base pairs for wrapping around the centromeric nucleosome. And that's only enough for a single wrap of DNA. We also were interested in the composition of the centromeric nucleosomes, so we pulled down with H2A, and we get a clear result here. You're looking at the superimposition for all 16 yeast centromeres, and we find that if we subtract input from H2A, we see that there's as much H2A over centromeres as there is genome-wide. And if H2A were absent, as some have claimed, it would be down here. So we see that basically the composition is all for, is basically, we see both the H3, that is the CSE4, it would send H3 or SEMP-A, and we also see H2A, so it's all there. However, I should point out that that's in vivo in yeast, but in vitro, you get a very different picture. So there's several papers that have shown you get canonical core particles in vitro by reconstitution, and some of them are in humans, some of them are in yeast. And Ramin showed that there's a high-resolution structure that shows an ordinary-looking core particle. However, it's only wrapped by 121 base pairs, so it's partially unwrapped. And that partial unwrapping is quite interesting because it implies that inherently that particle is less stable than a fully wrapped particle. And it's a point I'll get back to at the end. And the other thing is that when people have done this in yeast, people know what they're doing. They've been able to get it on artificial sequences, but when they try to use the natural yeast centromere, it doesn't work. If they use a centromeric DNA, it doesn't work. They can't get the particles, which I think is kind of a big problem here. So what I'm going to go through is how we might resolve that issue. In our own hands, when we tried this with Drosophila, we got right-handed particles, presumably hemisomes. But when we switched to yeast, we ran into all kinds of problems. We could get left-handed particles, right-handed particles. It depended on how we did the experiment. And our interpretation of this is as follows. The histone core is very basic. The DNA is very acidic. What's holding the nucleosome together is primarily electrostatic interactions. And those electrostatic interactions are going to be twice as many for an octamer as for a tetramer. So when we try to do these experiments where they're in competition, usually the octamer wins, and it's very hard to actually get anything like a pure tetrameric particle. So we decided to try a different approach. So we use an alternative reconstitution method, again going back to the 1970s, from Kelly Tatchell and Ken Van Holt in a paper that has never been cited, by the way. And what they showed was that if they used DNA duplexes that were about 65 base pairs in length, they could get octameric particles. And the idea is that actually there must be a gap in between the two duplexes, and then they can split them into two hemisomes in low salt. Now, the hemisome would be wrapped precisely by 62 base pairs of double-stranded DNA. So that's what we use in our experiment. So here's a gel shift in which we have a fully assembled left-handed H3 or CSE4 octamers plus 62 base pair double-strand DNAs. We go from high salt, dialyzed to low salt, and then we use a gel shift assay. So we see a gel shift for a sharp band here. There's also a fuzzy band here. And we see a moderately sharp band here for CSE4. CSE4 has a very long tail, longer than the core itself, and so it's going to run more slowly than H3. Now we can determine exactly the sizes of these particles using atomic force microscopy, which is very sensitive to particle heights. And what we see is that the height distribution for H3 is, there are actually two types of particles here and only one here, corresponding to what we see in the gel shift. And when we look at the length distribution using an internal DNA standard, so we can see exactly what the heights are, this corresponds to tetramer height, this corresponds to octamer height, whereas for this, it's mostly, almost entirely tetramer height. So what we're looking at here are tetramers. Now, we're able, because this wraps around in such a way that the two ends come close together within 50 angstroms, we can use a FRET assay, because we'll only get a FRET signal between the two ends, and we won't get a FRET signal for unwrapped DNA. So we excite with a blue laser. It emits in the cyan. It excites the other fluorophore, and it emits in the red. And we measure the FRET signal in the red and the donor emission in the cyan. And we can do this directly in the gel, because we can just superimpose the two scans. Oops. Okay, so here's what we see. So here are the bands I showed you before for H3. And we do this either with a fluorophore, a mixture of two equal amounts of two fluorophores on separate DNA molecules or on the same DNA molecule. So what we see for the tetramer, the H3 tetramer, is no FRET at all. And that's because there's only one fluorophore on the end of each molecule. However, when we have it on both ends, we see a nice FRET signal. And for octamers, some of the times, if there's two of them, they'll sometimes give you a FRET. They'll give FRET between them, so we see sort of a weak signal, but we see very strong signal when we have it on both ends. And when we do CSE4, we get pretty pure tetrameric particles that show nice FRET. We can then ask about the stability of these particles. So we use urea, because urea is a protein denaturant that won't interfere with the electrostatic interactions. We want to keep the DNA on there. We don't want to add salt and get rid of it. But the urea will separate the proteins. And it's quite effective. So when we go up to 1 molar urea or 2, the pseudo-octosome basically blows apart completely. And we just see the free DNA getting released. However, for CSE4, these hemisomes are really stable up to 4 molar urea, even up to 8 molar urea. It's there. We can't blow it apart at all. So we see very different behavior for the two. So the hemisomes are more stable than these H3 pseudo-octosomes. So what you realize is that if you just have a gap or a nick, a gap in the DNA, it's the DNA that's holding the particles together. You have a gap, and the protein-protein interactions won't hold it together, and it comes apart. But the hemisome doesn't have anything to hold together because it's nicely wrapped by the DNA. Now, what about the question of why can't you wrap natural yeast DNA, centromeric DNA around an octamer? So we decided, well, why don't we try this with the 78 base pair CSN4 sequence that I showed the data for. Since we know exactly where it maps, let's try this. And it works beautifully. So if we use CDE2 alone, what we see is H3 and CSE4 show very nice hemisomes. In fact, H3 is even better than CSE4. They're stable to urea, and if we cut the bands out of a gel, we see that all 4 histones are there. So it works both for H3 and CSE4, so you realize that what this region is, let me just back off. We're not getting octamers. We're only getting hemisomes here. We were getting octamers with this artificial sequence, but the real sequence only likes hemisomes. And I think what that's indicating is that it's not whether it's H3 or CSE4 that's important. It's that it's got to be a hemisome that's important. And so what this implies is that this CD2 region has evolved to basically exclude octamers and allow hemisomes to form. Now, what is it about CD2? Well, as Rick Baker had shown, it's got a preponderance, it's not just AT rich, but it's got a preponderance of runs of A's and T's. And it's known that runs in A's and T's actually make DNA stiff. And we can show that because if we look at the DNAs that have been used to wrap octamer particles, these are very bendy DNAs. But if we look at all 16 yeast centromeres, we get very stiff DNAs. And that makes a lot of sense if you think about it. If you wrap around the horseshoe shape of a hemisome, that'll go so far. But if you try to make that last turn to go all the way around, if the DNA is too stiff, it just won't work. So we think that that yeast CD2 has evolved to be so AT-rich with runs of A's and T's, essentially to omit, to prevent octamers from getting in. And it doesn't really matter that H3 can get in there as well, because CBF3 protein complex will recruit CSE4 to make sure that you'll always have high CSE4 there. Now, do octamers form in vivo? Well, here we did an experiment, actually quite by accident. We got a strain from my colleague, Sue Biggins, that we didn't know at the time actually had 5 to 6 copies of CSE4. So it was a slight overproducing strain. And what we noticed is that if we look at the length distribution, we had a peak at about 35 base pairs, 135 base pairs in length, which was different from the 155 base pair average size that we're getting from the same material pulling down with using H2A. And this turns out to be exactly the size as you would expect based on work from Kingston and coworkers, where they had assembled in vitro octamers, left-handed octamers, and they were able to use MNase digestion, and they basically showed these are the sizes. And so our interpretation is that simply when you overproduce the stuff, by mass action, you're getting octamers to form to go in. And where do they map? Well, they map actually throughout the genome. So if we line up all the genes at their 5' ends and then look at the median lengths of the fragments, we see an approximate 20 base pair difference between the sizes for CSE4 and the size for H2A from the same material. And if we map these at single base pair resolution in the genome, and we compare H2A to CSE4, we see that every nucleosome position has a little bit of CSE4. So it's going in at a low level everywhere. Although it's interesting, this comes from a paper from Ali Rando where he measured nucleosome turnover. So we're back to turnover. And what we see is that if we divide CSE4 by H2A profiles, we see that promoters show high, where they show high turnover, they also show a small abundance of CSE4. It's going in everywhere, but it particularly likes to go in here. And I think that sort of inspires the following kind of model for what's going on. What's maintaining a centromere at a single site? So because of the partial unwrapping, the CEN-H3s, we call these generically nucleosomes, go in, but they are they're more easily disassembled, as Ariel Purnell had argued. So they're more easily disassembled, so then they keep getting kicked out. So overall, there'll never be enough in there for a long enough period of time to actually form a kinetochore. However, at the centromere, we have the CD2 sequence, which is evolved basically to favor a hemisome, and we have CBF3, which is evolved to recruit specifically CSE4 as opposed to H3, so it will have high occupancy and it'll remain there. Now, to expand this to higher eukaryotes, where you don't have these proteins, I think we can consider the following, that as long as there's turnover, you'll be kicking out these centromeric nucleosomes that go in. But if you don't have turnover, you won't kick them out. They might go in, but they'll just stay there. And in fact, a defining feature of most centromeres is that they're embedded in heterochromatin. And heterochromatin is condensed, compacted DNA, in which there's really very little, if any, turnover. So we think that heterochromatin is an adaptation to maintain centromeric nucleosomes in these arrays and to prevent kicking out of these smaller particles. And with that, I'm going to just close and mention people whose work I talked about. Roger Deal, who has continued who did the work on Ketchet and is continuing in his own lab at Emory. Sheila Tevis, who did the heat shock study. Gabe Zentner, who did the iSwitch study. Taka Furuyama, Christina Krasovsky, and Christine Kodomo have contributed to the centromere work, and Georgia Henikoff did all the analysis. And I'll stop there and take questions. Thank you.
It's perfect timing. I'll begin up a question. So, Steve, when you use these the CDE elements, 78 base pair, did you chop down to see if smaller fragments also works well?
No, we did. We only did the 78. We have done other DNAs, but those are all based on the alpha satellite.
Okay.
So we've done different DNAs. 62 seems to work the best.
Hi, Steve. Nice talk. I have two questions. So you have shown very nicely that many histones are turning over very fast in certain regions of genome, therefore making the epigenetic inheritance very difficult. I'm wondering, since we know the epigenetic landscape in the genome is quite constant over the time, so do you know for the newly deposited histones, are they pre-selected to share the similar histone modifications as those that are removed, or they are actually, it doesn't matter what previously the histone modifications are, and then they will be modified to the same patterns? And the second question is, have you examined the histone turnovers across the different cell cycle stages? Thanks.
A different sequence?
Cell cycle stages, for example, mitosis.
Are you asking about? Oh, are you asking about the turnover?
Right.
Oh, well, the way we actually measure the turnover. So let me answer the second question first. The way we measure the turnover is that we're fitting an exponential decay model, and as a result, it really requires two events for us to measure it. As a result, it's unlikely that we're detecting any replication coupled assembly. It's almost entirely replication independent assembly. I think we'll be hearing more about this from Bing-Ju. So we think that what's dominating the turnover is replication independent assembly. Now, your first question.
So are the newly deposited histones, are they selected to share the similar patterns?
The modifications are newly deposited histones. So Genevieve El-Muzni has looked at that, and there are a couple of modifications that are pre-assembly, but most of the modifications that have been implicated in epigenetic inheritance are post-assembly. I think that's very interesting from the point of view, for example, of what we heard last night about ATRX and DAX, in the sense that if you turn over nucleosomes and they go in with H3.3 and they don't have any modifications on them, then whatever the enzymatic environment is around them, that will probably cause those modifications to go in. So I think we have a sort of a vague sense of how you might maintain modifications. You put in a naive form of H3.3, but it's going in the context of particular enzymes. And the case of ATRX and DAX is quite interesting because it's going in at largely at telomeres, which basically can put on the particular modification, one particular modification, which is H3K9 methylation. So therefore, H3.3 will get those modifications. As far as we could tell, all the modifications are the same on the two variant forms, different in abundance, presumably because of where they go in.
All right, thanks.
Steve, I have one comment and one question. So the comments regarding the spacing of nucleosomes maintained by the I-switch complexes. Actually, you mentioned that in the higher eukaryotic system, it may use the similar mechanism. So actually, we have data showing that BRG1 indeed is involved in that process. So if we inhibit BRG1, for example, the nucleosome spacing surrounding transcription factor binding sites did shrink. So the question is, you showed only the I-switch complex. Did you see risk or switch, Steve?
Oh, well, we haven't done risk. No, we've only done those three. Those were the ones implicated from other work, and we've only looked at those so far.
Okay, thank you.
Don.
Steve, you show quite clearly that your right-handed hemisome is really favored on the budding yeast centromere, and the octamer is not. What about, and I think you mentioned the Purnell evidence, and just if you could remind me there. So in the repetitive elements of the epigenetic centromeres outside of yeast, do those sequences also show that bias toward these?
We've looked, yeah. So first of all, we didn't show the handedness in vitro. So,
okay, I was giving you that.
We only showed the site.
Yeah.
Well, we don't know, actually. It's interesting. It might be flat, for all we know. With respect, we actually are using alpha satellite, so that's the natural centromeric DNA, although the way it's done for the octamers is a palindrome, and we're just using one part of the palindrome. We've used different lengths, and they all seem to work pretty well. Now, if we look at bendiness, they're sort of, oh, actually I showed, they're pretty bendy. So I'm not sure the rules hold. Let me just sort of point out a sort of a speculation here. I have a slide, actually. Yeah, so the idea here, so if we get to higher eukaryotes where there's these long arrays, and we have to realize that the alpha satellite includes all of this. So it includes the arrays of centromeric nucleosomes as well as what's around it. And you realize that it has to occupy the alpha satellite has to be on these nucleosomes as well as these. So, and our thinking is that because it's asymmetric and extended, that at mitosis, when there's all this condensation, these form sort of liquid crystals, and these get pushed out to the edge. So that's the way we're thinking of it. So we don't really think that the alpha satellite is about 2,4-SEMP-A. We think that it's actually about to make these really nice condensed nucleosomes, and that's why they make such good octamers. It's interesting that the structure of the H3 nucleosome was done on alpha satellite inverted repeat. So I think it kind of fits.
Last question.
So you show in the yeast the chromatin histone turnover is quite dynamic, about 1 hour. So what about in mammalian cell?
The turnover in Drosophila in mammalian, compared to mammalian cells, we're doing that. We're doing that right now. We can do the turnover data so we can do it in mammalian cells as well. Our problem is, remember what I was showing was particular epigenetic regulatory elements that have been mapped genetically and biochemically at particular sites in the Drosophila genome for trithorax and polycomb group proteins. But we don't really have that same kind of information, so it's hard to know what to line up. And you get a whole range, and so without knowing what to line up, I don't know. I don't know the answer to that.
Okay, thank you. So our next speaker is Craig Peterson. He's going to talk about nucleodynamics, put histones in place.
Put down a little bit. I tend to be loud, so maybe we'll put this down. Great. We'll start by thanking the organizers for the opportunity to speak and for the invitation to come to China for the first time. What I'll talk about today is a completely unpublished story. In fact, this will be the first time I've talked about it outside of our own group. It's a story that's a biochemical story, and it's dissecting regulation of histone turnover at promoters. So it's going to be a turnover talk. And if we think about the types of dynamics that occur in the context of transcription, or even in the context of repair or replication, or just dynamics in general, what we know is that basically dynamics can be separated into 3 basic levels. The level of the nucleosome is basically the dynamics of where nucleosomes are positioned along DNA. So which sequences are wrapped on a histone octamer obviously determines accessibility, and nucleosomes can move in cis. They can be mobile, moving around in cis. Nucleosomes, as Steve mentioned, also are not stable on DNA. They do have measurable, presumably, off rates where the entire histone octamers can come on and off, and newly synthesized histones can come back in, replacing or turning over the old nucleosomes. There's also dynamics, as we'll see, in terms of the H2H to be dimers, where dimers could come in and out, and it can be either replaced with the same dimer or histone variant dimers and so forth. And there's also dynamics at the level of the chromatin fiber, where these more extended structures are constantly being interchanged with more compact fibers. And of course, all these levels of dynamics are believed to regulate transcription. For today's talk, I'm going to focus on nucleosome turnover and mechanisms of nucleosome turnover at Pol II promoters. And we take a generic look at a generic Pol II promoter, and this could be yeast, it could be mammals. Essentially what one sees is nucleosomes, promoter proximal nucleosomes tend to be enriched with the histone variant H2AZ that flanks transcription start sites. And if you look at nucleosome turnover, so this exchange of histone octamers with newly synthesized histones, that turnover essentially occurs genome-wide, but it doesn't occur at the same rates genome-wide. Within coding regions, you see turnover of nucleosomes, and to see turnover at coding regions, that requires basically high levels of transcription to see turnover within coding regions. But what you see primarily are these hot nucleosomes where there's lots of turnover events. Very easy to see turnover at promoter-proximal nucleosomes. And that's true in yeast, it's true in mammals, true in ES cells, where these promoter-proximal nucleosomes show high levels of turnover. And importantly, turnover of promoters is not dependent upon transcription. In yeast, it's very clear that inactive promoters selectively have very high turnover of these promoter-proximal nucleosomes. And turnover at these inactive genes is thought to impact the kinetics of gene induction when you are going to turn that gene on. Now, if we look at the properties of nucleosomes that are undergoing turnover, all these nucleosomes, at least in yeast, and now there's some evidence emerging in mammalian cells, these nucleosomes harbor a histone modification, acetylation of K56 on histone H3. That marks all the nucleosomes. It marks the nucleosomes that are turning over very slowly or infrequently, and also marks the nucleosomes that are turning over much more rapidly at promoters. And what we want to understand is, why do these nucleosomes turn over much more rapidly? What's regulating the turnover of promoter-proximal nucleosomes? Presumably it gives the insight into, obviously, transcriptional regulation. What we know is that H3K56 acetylation, which again is a mark on newly synthesized histones, so it's going into these sites, that this mark is not just a passive mark that marks the turnover event, but in fact this mark is actually required for the enhanced turnover at promoters. What that means is that the first turnover event, where you introduce 56 acetylation into that nucleosome, that now drives subsequent turnover events. Essentially implies a positive feedback loop where K56 acetylation drives successive rounds of turnover. But obviously that doesn't really explain why these nucleosomes are targeted for turnover, because these nucleosomes presumably should also be going through that positive feedback loop, and they're not. So the only thing that's really special, that pops out is that these nucleosomes also have H2AZ in those nucleosomes. And in fact, it's also known that, so here's a positive feedback loop, but it's also known that H2AZ at these nucleosomes is also required for enhanced turnover at these promoter-proximal nucleosomes. So the question is that what's so special about this combination, H2AZ and H3K56 acetylation? Why does that drive enhanced turnover at promoters? And obviously the most simple interpretation, the simple answer to that question is maybe these nucleosomes are just inherently unstable. For whatever reason, they tend to fall apart, they're easily displaced, and so they're going through this turnover event because they're unstable nucleosomes. And there is some examples in literature where people have suggested that these nucleosomes are unstable. Mammalian cells, as I mentioned, 56 acetyl is probably in some of these nucleosomes. You can actually also consider this as being 3.3 as well. It marks the same kind of nucleosome. So we can test this very easily in vitro using recombinant histones, where we assemble recombinant yeast mononucleosomes in vitro, and those mononucleosomes either have H2A or H2AZ. And we use, in a lot of these studies, we use a mimic for K56 acetylation. It's where the lysine is changed to a glutamine residue. And there's good genetic studies in yeast saying that this, in fact, is a good mimic of a hyperacetylated K56 in vivo. We've used this primarily, though we have some work I'll show you on acetylated histones. We make these nucleosomes, we put a biotin group on one end of the DNA, we fuse these nucleosomes on the streptavidin beads, and then we simply measure stability of these nucleosomes using increasing salt washes and then Western blots just to do a quick measurement of how stable those nucleosomes are. And essentially what we find is that there's no impact of K56Q on stability of nucleosomes. And that's actually predicted by where that residue lies in the nucleosome. So here shows the data here for an H2A nucleosome with either unmodified K56 or K56 changed to Q. You can see here we're monitoring H2A, or the dimer stability. You have to go up almost a 0.9 molar salt before you start losing dimers from a normal nucleosome, and that's typical for nucleosomes. And what we see with H2AZ nucleosomes is these H2AZ nucleosomes are less stable than an H2A nucleosome. And that does make sense, because in fact, for yeast nucleosomes, yeast H3 is essentially equivalent to H3.3 of mammals. And there's been some indication from in vivo work that H3.3 H2AZ combination is unstable, and we now see that in vitro with recombinant histones, and in fact that dimer-tetramer interface is less stable, at least to salt. But when we make now the Z-Q combination, this novel combination, it's really not much more unstable than the Z nucleosome. So there's nothing magically unstable about a 56-acetylated Z nucleosome. Okay? So that doesn't explain why these nucleosomes are inherently unstable in vivo. And so we think about the things that affect chromatin dynamics. We know that histone modifications affect dynamics, and we have a histone mark, 56-acetyl. We know histone variants can affect dynamics, and we have our histone variant. These two are not sufficient, and that really leaves a third mechanism, ATP-dependent remodeling, as a mechanism to drive dynamics at promoters. I remind you that ATP-dependent remodeling enzymes are generally very large multi-subunit enzymes that hydrolyze ATP and use that ATP to translocate DNA, and as DNA translocases, they can move nucleosomes around and or disrupt nucleosomes. And these enzymes come basically in 4 flavors: switch-NIF class, I-switch class, the CHUD class, ENO80 class. Members of each of these families can use ATP to translocate DNA and to move nucleosomes around in cis. Only members of the switch-NIF class and the INO80 class can use that translocation activity also to disrupt nucleosomes, also either to evict histone octamers or to swap dimers around. So obviously these are better candidates for things involved in histone turnover. We've taken members of each of this group and asked, do any of these enzymes get stimulated biochemically by having H3K56 acetylation on their substrate? And I'm not going to show you the negative data, because essentially these enzymes that are just the sliders are not affected by K56 acetylation, whereas as these are. So I'll show you just a little bit of data. Here's basically a typical nucleosome eviction assay using the switch-NIF enzyme, where you feed switch-NIF a radiolabeled mononucleosome. In that reaction is also an acceptor bit of plasmid DNA. It's a very simple assay where you take switch-NIF plus ATP, switch-NIF evicts the octamer gets captured by the acceptor DNA, generating free DNA in the reaction. It's a very simple quantitative reaction. What you see here, when we feed it wild-type nucleosomes at this concentration of switch is a pretty lousy nucleosome evictor. Typically, the RISC enzyme is a much better evictor of octamers. Switch is not very good, but if you feed it now a nucleosome where K56 is mutated to Q, that gives about a 4- to 5-fold stimulation in the rate of octamer eviction by switch-NIF. We see about a 2-fold increase in the rate of octamer eviction by RISC. So at least at this level, K56 acetylation does enhance the ability of these enzymes to evict octamers, but doesn't really explain this kind of novel requirement for both Z and 56 acetylation, because Jerry Workman's lab has shown that, in fact, H2AZ incorporation actually inhibits remodeling by switch and RISC. So those enzymes don't really like Z. So this explains at least part of the data, but doesn't explain why you need H2AZ for this enhanced turnover. So we move to enzymes that clearly have a functional relationship to H2AZ, and these are the members of the INO80 family of enzymes. There's 2 flavors of these complexes in yeast. There's the SWER-C enzyme. This is the enzyme that does all of the H2AZ incorporation in yeast. There's a homolog of mammalian cells that does all the Z incorporation in mammalian cells. This enzyme in yeast is basically tethered to basically every Pol II promoter by sequence-specific DNA binding proteins. It's tethered, and that's why it basically puts in Z right at those promoter proximal nucleosomes. It's all about location and recruitment by binding factors. So what it does, it uses ATP to strip out H2H2B dimers and then replace them with H2AZ H2B dimers. So doing this dimer exchange reaction. INO80, we found a year or so ago, it does the counterpart. It basically recognizes Z nucleosomes that are in the wrong place, uses ATP to take out Z dimers, and puts back in an A dimer. So those 2 enzymes work together to maintain the proper localization of H2AZ within the yeast genome and likely also within mammalian cells as well. So we asked whether these enzymes are regulated by K56 acetylation, looking first at SWER-C. This is how we measure dimer exchange in vitro. We take recombinant yeast histones, we reconstitute mononucleosomes with those histones. We then react those with chromatin remodeling enzymes with free histone dimers in trans. So here's the substrate. We have the free dimers that are going to want to go in. And we take that reaction and we electrophoresis on native page gels to separate the mononucleosome product from all the other reactants. And that way we actually are guaranteed we're seeing exchange events. And then we Western blot that native page to ask what flavor of H2A is now in the nucleosome using different epitope tagged histones. So here shows kind of the scheme for what Z does. Here's a basic Z incorporation reaction by Swir-C. This is basically the reaction that was defined by Carl Wu several years ago when he identified this enzyme. Here's basically the Western blot for H2B or for H2AZ. You're starting with an H2A nucleosome, putting in Z. And as you increase Swir, you see increasing incorporation of H2AZ into this nucleosome. And I'm not showing the gels, but you also see the commensurate loss of H2A from that mononucleosome substrate. So that's with an unmodified H3 on an A nucleosome. Surprise was, when now we put in K56Q, we got a complete inhibition of the Z incorporation reaction. Single amino acid change in H3 somehow blocked the ability of Swir-C to basically take out A and put in Z. It's very surprising, considering think about this being a translocase dimer exchanging. And we wondered what was going on here. If in fact we thought perhaps it's actually doing the exchange reaction, taking out A, putting in Z, but when 56 is acetylated, it's now taking the Z back out immediately. So it's basically affecting the reverse reaction of this exchange reaction. So to test that, we basically fed Swir-C just an H2AZ nucleosome. Okay? Normally, Swir doesn't do anything to an H2AZ nucleosome because that's its product. It makes it, and then it leaves it and goes away, and nothing happens to Z when you give it Swir. If we take this same nucleosome and now we make K56Q, what we saw is a Swir-dependent eviction of Z from that mononucleosome substrate. Okay? So now Swir is basically evicting Z when normally it's wants to put it in, and again that's regulated by a single amino acid change in K56. In this experiment, it's K56 to Q. So now we look to see, does a single amino acid change, the acetylation event, basically switch Swir-C from an enzyme that's incorporating Z to an enzyme that now wants to incorporate A. So mostly doing lots of different types of dimer exchanging. So here's now feeding either Eno80 or Swir-C. It's feeding it here now with Z nucleosome with A dimers and trans. And then this is basically on a normal nucleosome, it would be Eno80 incorporating H2A into this Z nucleosome. These reactions were done at very low concentrations of Eno80, which is why you get such a poor incorporation here. You'll see in a moment it gets much, much better. But this is a typical Eno80 reaction. And now here's Swir-C on this substrate. Swir-C can't put in A no matter how much Swir you put onto this mononucleosome, and that's been shown by Carl Wu's group and other groups that enzyme on an unmodified nucleosome is really specific for H2A and putting in Z. But now we have K56Q. Look what happens. Now Eno80 is much more robust at putting in H2A. You can see eviction of Z. And now look, Swir-C now looks like an Eno80 enzyme. It's now taking out Z and putting in A. So now the Swir enzyme is now a promiscuous dimer exchanger, has no specificity whatsoever, seems to be both taking out A, putting in Z. As soon as it puts in Z, it takes out Z and puts in A. Okay? So it's kind of going back and forth, has lost its specificity for putting in Z. Now all these studies were using K56Q. So we want to make sure we're not just studying something odd about that Q residue. So we made K56 acetylation and tested that. So in this case, we're taking recombinant H3. We're settling it in vitro with purified the hat, RTT109, and its subunit VPS75. So this is provided by Paul Kaufman. We acetylate H3 in vitro. We then reconstitute octamers. We separate the octamers from the enzymes, reconstitute mononucleosomes, and do the same experiment. And luckily, we see the exact same thing. If we acetylate this nucleosome at K56, now Swir-C incorporates H2A into an H2AZ nucleosome. So now we have an example of a post-translational modification affecting the dimer exchange specificity of a remodeler. And taking an enzyme that normally shows great specificity and incorporating Z, when you acetylate that nucleosome, it now, we believe, puts in Z, but then also takes out the Z and puts in A. And when you're basically doing this histone turnover reactions, we have a lot of K56 acetylation around, we're basically getting, we think, continuous dimer exchange from the two different states. Z to A, Z to A, A to Z, Z to A. Back and forth, back and forth, back and forth. when K56 is acetylated. Now, one prediction of this kind of dimer exchange model to explain histone turnover kinetics is that if you look in vivo in strains where K56 is constitutively acetylated, one should see lower occupancy by chip of Z at promoters, because it's exchanging much more rapidly when you have this K56 acetylation constitutive. That's exactly what we see. If we look now at a variety of genes by basic chip in a K56Q strain, what we see is about 20 to 40% decrease in H2AZ in a variety of promoters when you constitutively make that K to Q. So again, that's reflecting, we think, this enhanced dynamics of Z at promoters. And then if we look at strains where we've now deleted both of the histone deacetylases for K56 acetyls, now we're getting basically very hyperacetylated 56 acetyl throughout the cell cycle, you get even more dramatic effects on Z occupancy at promoters. Furthermore, the prediction is if this dynamics is going on, then a K56Q mutant should basically behave genetically as an H2AZ hypomorph. It should look like you have low Z all the time. We've done now some gene expression profiles, at least preliminary ones, and what we find is that the gene expression defects in a K56Q mutant are positively correlated with mutants that lack H2AZ. Okay? So they seem to affect similar sets of genes consistent with Q, K56 acetylation, affecting Z dynamics at promoters. This then leads to the model for how you get enhanced dynamics at promoters. You basically have this situation going on. The first round of exchange generates a Z nucleosome with K56 acetyl. This novel nucleosome then directs SWIR1 to do a continuous promiscuous dimer exchange reaction where now these dimers are constantly shuffling back and forth from an A to a Z state, basically generating very unstable nucleosomes. And the data also suggests that these K56 acetylated A nucleosomes are also going to be acted on more frequently by switch SNF and RISC to evict those particles. So the combination basically says that Z, which we really didn't know why Z is at promoters, we think Z is at promoters because it basically directs these nucleosomes to be acted on by multiple remodeling enzymes, driving their instability. Now, the last couple of minutes, what I want to do is address the question, why does K56 acetylation impact the specificity of dimer exchange? Okay, so why does that affect that? And basically it comes down to looking at ATPase activity of SWIR1. Remember, the amount of ATPase activity of these enzymes directly correlates with how much DNA they translocate. SWIR1, it turns out, is a very lousy ATPase. It's about 50-fold less active than switch SNF. Turns over only about 10 ATPs per minute versus switch is about 500 or more per minute. And Carl Wu recently had to basically describe a fluorescence-based assay where you could quantify this very low level of ATPase activity of SWIR1. And basically what Carl finds is that SWIR enzyme has a very low basal activity of ATPase activity that's not stimulated by DNA. That enzyme is only stimulated by H2A nucleosomes. Okay? Its substrate. H2A nucleosome stimulates its activity, stimulates its translocation activity. And also if you now add Z dimers in as well, that gives you a further boost. Okay? Well, we decided to again reproduce Carl's fluorescence-based assay and measure ATPase kinetics, and we're able to nicely, so looking at rates here, it nicely recapitulates Carl's results. Here's that basal level, and H2A nucleosome stimulates it, and importantly, HTZ nucleosome does not stimulate it. For some reason, the Z nucleosome, which is normally the product, this enzyme cannot translocate DNA on a Z nucleosome. It's blocked. And we did the same experiment and just fed it K56 acetyl nucleosomes, and the shocker was on a K56Q nucleosome, there's now no specificity whatsoever. Here's the H2A nucleosome, here's the HTZ nucleosome. They all stimulate the activity of the enzyme. Okay? So what does this mean? It basically means that this residue is impacting how this enzyme sees DNA and how it translocates DNA to generate that remodeled state. I'll just end with a very fanciful model. Here's the nucleosome, here's H2H2B. K56 acetylation is right in this N helix of H3. It's kind of closely abuts the C terminus of H2A in this dimer. And essentially what we know is when SWIR comes in, SWIR sees this particle, hydrolyzes ATP, and based on other remodeling enzymes, we believe it pulls DNA in from the edge of the nucleosome towards the center, and that translocation reaction leads to eviction of H2H2B. Once it's evicted, SWIR1 usually comes with H2AZ dimer with it, or it can then plug it in. Once it plugs it in, though, it now does not want to take it back out again. And what we know is that there's a subunit of SWIR complex called SWIC2. SWIC2 binds to H2AZ, and it binds the Z right at this region here next to this N helix of H3. And we know that SWIC2 is required for stable incorporation of Z into nucleosomes, and we think it actually prevents the removal of Z. And the model, as we think, is that based on the proximity of these components, that K56 acetylation basically breaks that lock, does not allow SWIC to protect that Z nucleosome. We now get ATP hydrolysis in that case, and that leads to eviction of Z, and now it can do both reactions, putting in the A. Okay? So we have a modification affecting, we think, the ability of a specific subunit to basically drive the specificity of the reaction. This predicts that INO80, which specifically does the other reaction, is going to have a subunit that specifically binds to the H2A dimer and blocks that back reaction as well. So I'll just stop there. Acknowledge all the work I told you today was done by one very talented chromatin biochemist, Shinya Watanabe, who's studying back here. And of course, this is all done at UMass Med. Any questions?
Thank you.
Steve.
I wonder if the interaction is with K56 is actually with DNA gyres and having some effect there, and that's what's unlocking, as opposed to some direct effect.
Yeah, I mean, K56 acetylation, if you look on the structure, I mean, K56 acetylation does make a DNA contact, but as you know, it's a very weak hydrogen-mediated contact. It's right in the middle of a big basic patch where there's a lot of arginines and lysines that make more stable contacts with that, the entry DNA there. So most of the work that's been done in vitro on K56 acetylated mononucleosomes, you really see no change in nucleosome structure, no change in wrapping, other than a slight increase in mobility at high temperatures. So I don't think it does anything direct. We think actually it's the flexibility of the N helix of H3 that's impacting this. And it's actually, we think, the interaction between the N helix of H3 and the H2AC terminal domain, those interactions we think are what's key. So some data saying if you delete the C terminus of H2A, that also seems to affect the ability of SWIR to keep Z into a nucleosome. So I think it's that kind of intranucleosome. Yeah.
Hi, Craig. It's a really good talk. So, looks, the H2AZ, dynamically, swap in and swap out on the promoter region by, AD and SWIR1. So, do you think the H3K56 acetylation is a key for, switching, this, event?
So do I think at K56 acetylation?
Yeah.
Yeah, I mean, I think you need that combination. I think it's because the Z nucleosome by itself is happy, it's stable. SWIR is putting it in, but then it doesn't come back out again. I think it's really then that first exchange event now makes it a Z with 56 acetyl, and that's what triggers this whole response. Okay. So it really is that positive feedback loop triggered by the acetylation. Okay.
How they, the SWIR1 and AD can switch, to these two different events.
So how do they switch those?
Yeah.
Well, I mean, I think the key really is that acetylation event affects how this enzyme behaves on the different substrates, right? So basically that acetylation event basically allows SWIR to do the back reaction that it normally doesn't do, right?
Okay.
In a minute, I'll get the question.
Last question.
Can I ask about what happened during DNA damage?
Doing DNA damage. So doing DNA damage, which we've done a lot with, right? You make especially double-strand breaks. At double-strand breaks, you also see very rapid recruitment of SWIR and INO80 to breaks. Stefan Yanks has shown that as soon after you make a double-strand break, in the first 30 minutes, you see very dynamic incorporation of H2AZ at double-strand breaks. We've shown that INO80 is also involved in removing that Z later on, which regulates the checkpoint. And we know that incorporation of Z at breaks is very important for resection at double-strand breaks. Actually, at the Shanghai meeting last week, I talked about that story. It's not clear that there's a role for 56 acetylation at breaks. At one point, we wanted to look at turnover at breaks to see if turnover was enhanced at breaks, but turnover happens so well throughout the genome in yeast that we couldn't see any increased turnover at double-strand breaks. We don't know if this reaction, this extra cycling happens at breaks. It could.
Thank you.
Okay, thank you. So the talk before the break is by Zhi Guozhang from Mayo Clinic. He's going to talk about histone H4 phosphorylation and HER-A and CAF1 dependent nucleosome assembly.
So let me first start by thanking the organizers to give me this opportunity to talk about our work. My lab is mainly interested in how chromatin states are inherited during S phase of the cell cycle. More specifically, we are interested in how nucleosome assembly pathways are regulated. So today, I'm going to talk about how regulation of replication coupled to nucleosome assembly in yeast by histone acetylation. I'll give you a brief outline what we've learned from that, and then I'll tell you a brief story about the phosphorylation of histone H4 regulates nucleosome assembly processes in mammalian cells. So this is over a simplified view of replication form. This represents one nucleosome. In order for replication machinery replicate this nucleosome DNA, this nucleosome must be temporarily disrupted. Immediately following replication, two process replicated DNA is assembled into nucleosome through two processes. First is the transfer of parental histone H3H4 tetramers behind the replication fork. The second is the deposition of newly synthesized histones onto replicated DNA. While it's still not clear how parental histone H3H4 are transferred, it is known that the position of newly synthesized histones require histone chaperones, including CAF1, ASF1, and RTT106. In the last several years, studies from many labs, including ours, suggesting that histone acetylation plays a really important role to regulate the low-fold nucleosome assembly. Briefly, that the newly synthesized histone H3, H4, which is unmodified, first bind to histone chaperone ASF1. ASF1 then present the histone acetyl transferase complex RTT109 PPS75, which acetylate a specific lysine residue, H3 lysine 56, which you heard from Craig a moment ago. Once lysine 56 acetylated, it will transfer to two other histone chaperones, CAF1, RTT106. And these two histone chaperones then deposit histone H3H4 tetramers onto replicated DNA to form nucleosome. So there is, at least in this process, we believe there's a switch from dimer during newly synthesis and to tetramer during deposition. So what is the functional implications of the deposition of H3H4 tetramers by RTT106? So studies from Drubin's lab suggesting that three types of nucleosomes can be formed during S phase. That is, nucleosomes formed using parental histone H3H4 tetramers, new H3H4 tetramers, or mixed H3 tetramer containing one dimer of parental H3H4, one new H3H4. The position of H3H4, new H3H4 tetramers by RTT106 effectively inhibit formation of mixed, so-called mixed nucleosomes. We believe this is a way to regulate how nucleosomes are assembled during S phase of the cell cycle. So from all these studies, we learned that modifications on newly synthesized histones can regulate the low-fold nucleosome assembly. So we would like to ask whether this idea can also occur in mammalian cells. So I must say that most of histone chaperones are conserved from yeast to mammalian cells. There's one major difference between yeast and mammalian cells. That is, yeast has only one histone H3, but in mammalian cells, there are canonical histone H3 for the 3.1 and 3.2, and the histone H3 variants, which is we introduced last night and today by Steve. And 3.1 and 3.3 differ only by 5 amino acids. However, 3.1 and 3.3 are assembled into nucleosomes from different nucleosome assembly pathways. 3.1 is assembled by Cap1 in a replication coupled nucleosome assembly, and 3.3 is assembled by HERA and the DAX, which was introduced yesterday by Dinsha, in a replication independent nucleosome assembly. Previous studies of mutational studies from Steve's lab, Steve Henikoff's lab, indicate that 3.1 and 3.3 specific residues are very important to specify how this 3.1 and 3.3 are assembled. We ask whether any modifications on histones can regulate these two processes. To do this, we purified the ASF1A and ASF1B complex from 293 T cells. This is mock, and identified modifications and co-purified histone H3H4 by mass spec. One modification we find is phosphorylation of histone H4, serine 47. So this phosphorylation of this residue was described in the literature before. However, the function of this modification was not known. So we generated antibodies recognize this modification. And you can see that our antibody recognizes H4 in 293 T cell extracts, but not recombinant H4. Using this antibody, we test how PEG2 phosphorylated H4S47. I must say that in the literature, in the very old literature, it is known that PEG2 is H4 kinase. So, however, the specificity of this kinase was not tested. We purified the PEG2, wild-type PEG2, and the kinase dead mutant from 293 T cells and used them to phosphorylate 3 different substrates, mononucleosomes, 4 histones, and H3H4 tetramers. You can see that wild-type PEG2 can phosphorylate H4S47, but not kinase dead mutant. And this phosphorylation only can occur when core histones or H3H4 tetramers were used as substrates, but not mononucleosomes. These results suggesting that phosphorylation of histone H4S47 occurs prior to assembly of H3H4 into nucleosomes. Next, I will show you several experiments to address the function of this modification in nucleosome assembly. One of the experiments gives us a clue the function of this modification is we analyze the level of H4S47 phosphorylation in purified 3.1 and 3.3 mononucleosomes. This is loading control. This is the flag tagged 3.1 or 3.3. These are the core histones. This is Western blot of H3. You can see that H4S47 phosphorylation is enriched on 3.3 mononucleosomes compared to 3.1 nucleosomes. These results suggesting that this modification may be enriched on 3.3 enriched genes. To test this idea, of course, we are using chromatin immunoprecipitation assay. Shown on the left is the H4S47 phosphorylation chip assay in non-targeting or PEG2 depleted cells. It is known from KG's lab that these 3 genes are marked by 3.3. These 2 genes are marked by 3.1. You can see that more H4S47 phosphorylation were detected on these 3.3 enriched genes compared to 3.1 enriched genes. Moreover, depletion of PEG2 results in significant reduction of H4S47 phosphorylation. These results suggest that PEG2 is a major kinase for H4S47 phosphorylation in cells. And more importantly, we asked how depletion of PEG2 affects H3.3 occupancy. You can see that the depletion of PEG2 results in significant reduction of H3 occupancy at the candidate gene tested. In summary, what I have told you that H4S47 phosphorylation is enriched on 3.3 containing mononucleosomes, is localized at the candidate genes marked by H3.3, and the depletion of PEG2 results in reduced H3.3 occupancy. Expression of phospho-MYC H4S47 mutant has opposite effect on H3 occupancy as PEG2 depletion. I don't have time to show you these results. From these results, we suggested that PEG2 and H4S47 phosphorylation promotes nucleosome assembly of 3.3 H4 tetramers. So next, we ask how does PEG2 and the H4S47 phosphorylation does this? So there are two possible models. One model is that this phosphorylation will affect the binding of H4 to 3.1 and 3.3 differently. The second model is this phosphorylation will affect the binding of 3.1 H4 and 3.3 H4 tetramers with the corresponding histone chaperones differently. Through a series of experiments, I don't have time to show you. I can show you. We can say that this is unlikely to be true. I show you one piece of data supporting the model that H4S47 phosphorylation can affect the binding affinity of histone 3.1 and 3.3 with the histone chaperones differently. So here is IP Western blot experiments. We purified IP H4 or H4S47 mutant from 293 T cells. This is a loading control. You can see that significantly less CAF1 co-purified with E mutant as detected compared to Y-type H4 as detected by both subunits of CAF1. In contrast, significantly more HERA co-purified with H4S47 mutant than Y-type H4. These results supporting the idea that this phosphorylation can increase the binding affinity of HERA with 3.3 and H4 and reduce the binding affinity of 3.1 H4 to CAF1. So we can show that, in summary, we can show that the PEG2 can phosphorylate H4 complex with 3.3 or complex with 3.1. At this stage, we can show that there's no discrimination. However, once H3.3 H4 is phosphorylated, it will increase the binding affinity of HERA, which in turn promotes replication independent nucleosome assembly. And then with this, with 3.1 H4 is phosphorylated, it will inhibit the binding of CAF1 with 3.1 H4, and then inhibit replication independent nucleosome assembly. These results suggested that in addition to 3.1 and 3.3 specific residues that can regulate nucleosome assembly pathways differently, we think phosphorylation of H4S47 provides a lot of means to regulate these two nucleosome assembly pathways differently. So I would like to thank the people who did the work being, and we have a lot of collaborators on various projects, and we would like to get the support from the different funding agencies. Thank you.
Thank you. Any questions?
Yeah, just one question. So you show that the assembly of the histones into the nucleosomes inhibit the phosphorylation events. And I'm just wondering if you include the ATP remodeling complex in the reaction that could allow the phosphorylation of S47.
So we did not do the experiment. We don't know. So, but we know that this phosphorylation, if you occurs even in cytosol. If you can do self-recognition, H4S, you can detect H4S47 phosphorylation occur in cytosol. This suggesting that maybe one of the function of this modification occurs in the cytosol prior to the assembly of H3H4 into nucleosomes.
Okay, thank you.
Do you see any cell cycle effect by knocking down PEG2 or replication defects? So
far, we locked down PEG2 in different cancer cell lines. It appears that it's cell line specific. In HeNa cells, and the 293 T cells we used to do our study, there's not much effect on cell cycle or growth. However, on certain kind of cell lines, and it has growth defects.
I see. So that might have like cell-specific effect on like.
Yes, yes.
Okay, we'll take a 20 minutes break, and please come back by 10:50.